Hi guys. So thanks very much, Stephen, for asking me to come here. And I'm keen to talk to you a bit about, I suppose, what I do, which is a bit unusual. I guess I've described it as interfacing biology and technology, which effectively means I do neither technology nor biology particularly well, which might summarize it quite well. I'm based in the Department of Plant Sciences, and actually my background isn't biology at all. It's physics. So I did my undergrad and PhD in physics. Oh, lovely, thank you. Yeah. Oh yeah, sorry. I like to wander around, so my apologies. So I'm based in the Department of Plant Sciences as what's called an interdisciplinary fellow, which kind of permits this, I suppose, dual life of engineer, physicist, and biologist. So, I suppose, my perspective on things is maybe a little bit unusual. So just to kind of give you an idea of, where people thought we'd be around 20 years ago. So this is Shiro, this is the ghost in the shell. People really thought that we would have biology and technology truly integrated and interfaced. Kind of an ability to sort of meld between one and another, And I think the answer is that we're a long way from that. You look at any of us, and even those that, use prosthetics or anything like this, we really haven't gotten that far from 20 years ago or even in the last 50 years. And I suppose this is another example of kind of where people thought we'd be. So if anyone's familiar with this is a tricorder. This is a Star Trek invention dreamed up way back in the 1960s. And the idea is that this is something that, an imaginary instrument that can, measure anything. It can effectively test illnesses, detect atmospheric conditions, geological samples. And I suppose effectively it's kind of like a communication method, based on, what biology communicates with, which is, chemical signals. And I suppose, when you compare it to, where we've sort of gotten with, conventional communications, right? I suppose I can, with a few touches of a button, have a conversation with someone on the other side of the world. Or, for instance, share what I'm doing with, millions or even billions of people, on a network. And you can even look at this device and say, they were ambitious in their design. A TV on one of these units. What an incredible concept, But I mean, even look at the screens that we experience today, the size of our devices. They've really gone way further past that. But the actual biological components, the measurement instruments, the technology surrounding that really hasn't gotten where people thought it would. And I suppose to kind of give my interpretation of what the problem is, biology is beautiful. I think we'd all agree with that. It's very diverse. But I suppose in that beauty and diversity comes complexity. And really complexity at almost all scales, Regardless of what scale you're looking at, there's more and more information available. And our ability to control biology has indeed improved. We have methods to control, manipulate, and engineer biology. But I guess the question is, has it really advanced, all that much? And I guess the answer is, look at these tools of biology. So, you have freezers, growth medium, chemicals abound, in order to control and manipulate this biochemical environment and, control this communication method of cells and biology. And you can see there pipettes and just instrumentation that really hasn't evolved a huge amount in 50 years. So, that kind of brings me back to where I'm coming from. So this is the whole idea of this technologist in a biolab. So, these are tools that are more familiar to me. So you can see stuff like computation, engineering tools, electronics, optoelectronics. My particular interest is optics, and, fabrication methods. So these are the kind of tools that are, and instruments that are a bit more native to me. And, another kind of, I suppose, an abuse of concepts and an abuse of biology is that, I very much think of, biology in terms of, I suppose, an engineer. This kind of concept that really a cell or any biological instrument is sort of nothing more than, a set of components, that are integrated together to perform a specific function and controlled by, let's say, a central processing unit that is commanded with a language, written in DNA. So that's ultimately, where I'm thinking of biology. And, I suppose one of the issues here is actually the complexity of biology, if we were to put it at a sort of, computational analogy, is something akin to more like one of these huge Google data centers of several football fields, right? So imagine you walked into a Google data center, no one with you, and you didn't know the language that this architecture was controlled in, nor the methods in which it was connected together. That's effectively what you're kind of looking at with biology, that level of complexity and that level of vastness. And it's really difficult to engage with that. So I suppose what my research does is very simple. I break these cells down. What don't we need? Well, I don't understand the cell wall. Let's dump that. All that central processing unit, get rid of that genomic DNA. Effectively, I want to kind of go back to the sort of Raspberry Pi of biology, if that makes sense. So I work on systems called cell-free expression or transcription and translation systems. And, what's beautiful about that is that we can really begin to start to think in terms of, electronic components, that are, uniform, linear, operate in a specific design set of parametric ways, and then we can start to communicate that into stuff that we've observed and can observe in biology, simple processes that can kind of be adapted and sense things, So, think for instance, in electronics, switches, oscillators are core components. We can break those down into biological elements, and then we can go further and actually engineer these components using synthetic biology. So my research really thinks of, what's the simplest thing you can do, and how can you do it? So I'll sort of take a little bit of a diversion here, and again try to explain it in the way that I understand. Is anyone familiar with this particular image? So I sort of give you a hint now. You have, everybody here would have at least one on them, or probably more like several million or billions, right? So this is actually the first transistor. You can see it's scale. It's 2 centimeters or so across, and you can see that they've crudely taken a razor blade onto a piece of germanium semiconductor substrate and attached a few paper clips in order to produce something that gives this effect of changing one voltage when there's another available. So this is kind of the sort of thing that really inspires me. And through my career, I was lucky enough to join Toshiba as a postdoc, and what we were focused on was taking, instead of this, electronical control method, right? We were interested in using photonics as our method for integrating our transistor. And when you think of it, it makes a lot of sense, because ultimately nowadays, right, we pump photons or particles of light down optical cables rather than pumping electrons down cables anymore, right? So we were very interested in integrating photonic elements or photonic transistors into our devices. And again, to do this, it comes back to the advancement of hardware and the advancement of technology surrounding these components. So I showed you this, image of a transistor, a rudimentary, very rough component, and gradually that's defined using, more and more advanced tools and hardware surrounding it in order to integrate it onto a single chip. So this is a good example of that in a photonic analog. So this is something that controls the movement of photons around a microchip. And again, this is something that we tried to integrate and command and control on microchip-sized elements. So, this is something that's, a few nanometers in diameter and several microns high, and our focus was integrating that on chip. So that's really what kind of gets me going, being able to, take a technology that's, I suppose, at a reasonable early stage and really messing around with it, manipulating it, and trying to control it, and ultimately, make something productive and useful out of it. So that diversion now brings me back to biology and what my interest is in it. So I've told you that, we can make very basic and arbitrary circuits in biology, but what I think is lacking is this surrounding hardware. I showed you the tools earlier that we use to do these sort of manipulations, and they are really rudimentary and arbitrary, So, what we did a couple of months ago, and this is an example of a few projects that were conducted during this event, we ran a biohackathon with a kind of emphasis on people going nuts and building tools and open source technologies in order to explore and just try and do these things that engineering does very well. You know, manipulation, control, miniaturization. And this is a good example of that. So this is fabricating a mold that ultimately is quite small, and is going to be a mold that will be etched out. So it's placed inside a mold mask. You can see this mask here. And then subsequently, we're able to manipulate fluids in that device after we etch out that material. So now you can start to think that, you know, if you're really going to do biology, rather than moving fluids around with a pipette, it's very nice to consider moving them around using a fluidic device. And although this is very rudimentary, you know, I would hark back to that kind of transistor concept, that it is a turning point. It's something that, you know, allows us to go a little bit further forward. Obviously needs 50-odd years of further development, but it's getting there. And then this is this device embedded in a simple, open source, you know, conventional electronic system. All of this kind of amounting to around 30 pounds, the majority of which being that, you know, Raspberry Pi there that's running things, the 25-pound component. And I think another feature that is very important and was demonstrated during this particular event, and this is a very nice project example of that, is that, you know, the language and architecture of biology really does need to change, right? It's so complex that we need to communicate it in a, you know, simple, easily articulated manner. And that's something where, you know, having an online design facility and perhaps an exchange method and finally an automated cloud-based laboratory is very useful. So to give you an idea, I think I can play this. To give you an idea, a group that we were working with developed this online portal which allows you to develop organisms. In those organisms produce arbitrary circuits, so you can see that this is sort of the language of biology, right? You know, promoters, ribosomal binding sites, genes that encode for different things, and ultimately being able to send that off, and rather than doing any of this, you know, manual pipetting, really going away and automating the hell out of this process, getting people to manipulate and move robots around for you, so that you're able to focus on designing rather than actually fabricating and producing. And this was Transcriptic, this fantastic cloud lab in California, in San Francisco, that sort of facilitated this. And just to acknowledge the people that actually did this fantastic work, this is this Linkalab, this is this wonderful microfluidic tool. So again, that focus on miniaturization. And these are the chaps that did this amazing online portal, you know, a focus on automation. And, you know, overall, this wonderful event was conducted over 3 or 4 days, and you can see the scale and the passion that people sort of come with, you know. So 50-odd people come together and try to, you know, maneuver and manipulate things that, you know, can kind of create a new set of tools around biology in order for, you know, I suppose, what would you say, really, the development of new technologies, new biological technologies. And, you know, over the last couple of months, I've been privileged to be invited to be, an advisor to this amazing initiative, the Biodesign Challenge, which is going to be conducted in collaboration with the Royal College of Art. Now, what's brilliant about this is that it really focuses on, you know, saying that the science exists, you know, the science is here. You know, how can we structure, how can we create around that? So when you think of, you know, innovators and conceptualizers like, you know, Apple, you know, or you know, fantastic Silicon Valley innovations, a lot of the time it's not just the hard science that's important. It's really the way it's packaged, the way it's you know, put together. And I think designers are sort of the under-recognized geniuses of this, you know. I think a lot of the time, you know, the scientists get fantastic credit, and sometimes, of course, well-deserved. But I think it's important, if we're going to make things that people can use, that we really engage and sort of break disciplinary boundaries. And that's really what this challenge is all about. It's about taking designers, putting them completely outside their comfort zone in the world of science, and pulling science into that design cycle, so that at every step there's a sort of a design-informed process. And this is a brilliant international initiative that, you know, I'm delighted will be happening. And it's coordinated initially by GenSpace, who unfortunately their logo isn't up there, but a brilliant open source biomake space in New York, where the finale will be in June. So, you know, I'd encourage you to keep an eye on this. And finally, I'd just like to acknowledge this, this great lab I've been delighted to be a part of. And to kind of give you, a sort of summary view of it. What's quite amazing is that, ultimately it's best described by, the ability of biology to sort of travel through time and space, right? So this is the symbol of our department, this beautiful ginkgo leaf, and ultimately it comes from 1896, so over 120 years ago from Montpellier. So it's travelled, across the earth through time in order to join us in our department. And it really does sort of summarise, I think, what's incredible about biology. And I'd just like to acknowledge my fantastic laboratories. I'm delighted to be a part of the synthetic biology lab and plant sciences, and the animal technology lab of Lisa Hall, and all the fantastic people that I've had a chance to work with, and thank them.
Yep. Okay, any questions, sir? I have a microphone.
Thank you. Hi, very nice work. Just want to ask you, you really think that there is a sort of analogy between the optoelectronic world and the biological world, but there are also some differences. Is those differences in a sort of more random type of distribution of messaging, or is it one is soft matter, it is hard matter? Can you comment about that, please?
I think actually you're dead right. In a lot of ways, it's a terrible analogy. So ultimately, every process is different, and I suppose the complexity of biology is such, and it's kind of advanced above the scale, really, like of this sort of, at electronics or in optics, we're dealing with solid state components, where we can have a grasp over these individual particles that are doing things and operating in definable ways. I suppose, like you say, there is a randomness in biology, right? And it's at a higher scale, so you have multiple different interactions between chemicals and everything like that. So I suppose it is a very poor analogy, but I suppose it's kind of the only one I can draw upon. But yeah, I hope that in somewhat answers it. Yeah.
So, Thomas, I mean, for you in particular, is there something that, given a lot of resources, that something probably a bit more specific you tell us that you'd really like to do now?
Yeah, I mean, I think in the course of all of this, what's particularly interesting to me is really breaking it down to the very simplest elements. And I suppose the simplest elements are very basic biological circuits, and thinking, can we begin to create realizable, simple sensors using those? And if you can start to interact with biology, so sensing and having a response, you're very much getting close to the same concept in electronic sensing or electronic measurement. And I think that's where we might get towards these cyborgs, when you're able to control and engineer biology in such a way that you can get a cause and effect. That's when you can start to think of biometric patches and additional prosthetics that are sort of advanced beyond what we have available to us. So yeah, I think that's kind of what I'd love to see, I suppose.
Okay. Any more questions? No. Okay, we have one back here. Sorry.
Yeah, coming from physics as you do, going into biology, what did you find was the one sort of idea or sort of tool that you brought that biologists most appreciated?
I suppose the truth is, do they appreciate any of it? Maybe not. I think a lot of it is. I suppose that's a good question. Yeah.
What's the one thing you wish they appreciated then?
To be honest with you, I've kind of been more. I think I've taken more than I've given, in the sense that I love all this cool stuff biologists do. Everybody has even their world is orbits around a particular organism, and their expertise are so detailed. the way that they do things can often be so diverse. I mean, I think, I suppose really I would just think that automation is something that probably should be, you know, top of the list maybe of things that could be embraced more, perhaps. Yeah, that might be, to start a roundabout answer your question.
Yeah, thanks.
Do you think there will be applications of your expertise in nanotechnology and maybe seeing how nanobots can communicate with each other in a mesh network or something like that?
I suppose I suppose that is yeah, a nice concept. I don't know really. Yeah. But what do you mean by that? Kind of.
I mean, you're talking about the convergence of biology and technology in the future. You know, as described 20 years ago, etc. Hoping it will be today, but even if it is a bit late, I suppose eventually we might come to a point where we have nanobots swimming around in our bodies. So there's this sort of comparison of cellular components with electronics and optoelectronics. Use some of those sorts of knowledge and skills to create maybe a means for nanobots to communicate with each other.
I mean, I suppose in some ways, you know, we do have those bots roaming around us, right? We have, you know, microglia, macrophages roaming around our bodies, and you know, in some ways, like, why build it from the ground up when it sort of already exists, and perhaps you could interact with it, and you know, I guess that might be what I would wonder. Yeah, I don't know. I suppose you could. Yeah.
Okay, I'll pass it on to Stephen.
Thank you very much, Thomas. That was very insightful. So, guys, we're going to go on a quick break. So we'll be back at 11:30 for our regularly scheduled